Hello everyone, have a blessed day. This is Richard Devera, and we are going to discuss about the process of mitosis and meiosis. First, you have to define what is mitosis. Mitosis is a process where a single cell divides into two identical daughter cells. It is conventionally divided into two stages or phases known as interphase, prophase. In interphase, this is the longest stage of a cell's life, the period of the cell cycle during which the nucleus is not undergoing division, typically occurring between mitotic or meiotic divisions. The cell cycle of interphase is part of interphase is also known as the G1 phase of the cell cycle. This is the growth phase. And the second part of interphase is known as the S phase of the cell cycle. This is the synthesis phase when DNA duplicates. And the third part of the cell cycle is the J2. It's just a checkpoint to make sure the DNA is correct. And the last part of the cell cycle is called mitosis and has 4 phases during which the cell divides into two cells. The 4 phases of mitosis. The first phase of mitosis is the prophase, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, condenses. In mitosis, the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During metaphase, the cell's chromosomes align themselves in the middle of the cell through a type of cellular tug-of-war. The chromosomes, which had been replicated and remain joined at a central point called the centromere, are called sister chromatids. Long protein filaments called kinetochore, microtubules extended from folds on either end of the cell and attached to the kinetochores. In anaphase, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Before anaphase begins, the replicated chromosome called sister chromatids are aligned along the equator of the cell of the equatorial plane. The sister chromatids are pairs of identical copies of DNA joined at a point called the centromere. The separated chromosomes are folded by the spindle to opposite poles of the cells. And the last and the final phase of mitosis is the telophase. This is the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Telophase begins once the replicated paired chromosome had been separated and pulled to opposite sides or poles of the cell. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. So that's the mitosis. Now we are going to what is meiosis is all about. Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our 6 cells, the sperm in males, the eggs in females. In meiosis 1, the first of the 2 consecutive divisions of the nucleus of eukaryotic cell during meiosis, and composed of the following stages: the prophase 1, the metaphase 1, anaphase 1, and telophase 1. In prophase 1 of meiosis 1, the starting cell is diploid, and this is the 2n equals 4. The homologous chromosomes pair up and exchange fragments in the process of crossing over. In metaphase 1 or meiosis 1, the homologous pairs line up at the metaphase plate. In anaphase 1, homologous separate to opposite ends of the cell and the sister chromatids stay together. In telophase 1, the newly formed cells are haploid. This is the n equals 2. Each chromosome still has two sister chromatids, but the chromatids of each chromosomes are no longer identical to each other. In meiosis 2, the second of the two consecutive divisions of the nucleus of eukaryotic cell during meiosis, and composed of the following stages also: the prophase 2, the metaphase 2, the anaphase 2, and telophase 2. And prophase 2, starting cellular, the haploid cell mid and meiosis. This is where in meiosis 1 came from. And the chromosomes condense and the nuclear envelope breaks down. The centromeres move apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes. In metaphase 2, chromosomes line up at the metaphase plate, or the tug of war. In anaphase 2, the sister chromatids separate to opposite ends of the cell. They are already divided. And the last phase of meiosis 2 is the telophase 2, and where the newly forming gametes are haploid and each chromosome now has just one chromatid. And that's it, everyone. Have a blessed day. Thanks for listening or watching, and I hope you learned something about our topic. Thank you.